especially looked upon highly is because there's this real romantic notion that ring species are demonstrations of the process of speciation, like the temporal process, but realized in spatial dimensions. And so an example is the salamander species that is in California, and it's colonized around the Sierra Nevada Valley, and differentiation is due to trying to avoid predation, and it's such that they spread southward along the two ends, and there's lots of degrees of isolation at this terminal end here. So one of the observations is that there are relatively few observed ring species relative to the number of possible geographic barriers that would be capable of supporting, right? Example is in this paper from 2012, they took two possible sort of ring species barriers, looked at elevation gradients, and they came up with a large number of geographic barriers that could potentially support ring species that had similar characteristics. But the thing is that there aren't that many ring species. So, I mean, there's the one sort of obvious reason. The reason is that people don't look too much. One of the reasons why people aren't looking that much is because frequently in the literature, if they don't go back to the ring species, don't really follow strictly the romantic notion of the temporal and spatial processes of speciation. But I will present a model that shows a different possible mechanism that could explain this. And so that is what the objective of the model is, to provide this mechanism. So I tried to keep it as simple as I could. So you have two loci, an A and B loci. If you're suboptimally adapted, you have no local adaptability, you're lowercase. And if you are locally adapted, you're in uppercase. And the lowercase are the ancestral forms, and the uppercase are the derived alleles. So for example, if you're all lowercase, then you're just suboptimally adapted. If you have the big A alleles, then you're locally adapted. And then we set up this fitness model where the additive should be over there, but I made a mistake. But you have it so that we create the model so that it's a competition mediated process, and we use the Volterra model. So you have your R and your K. And the K is the carrying capacity, which I hope everyone should be familiar with. R is the intrinsic growth rate. So if you're locally adapted, your carrying capacity that you can experience goes up. So you're better at dealing with competition, but your intrinsic growth rate goes down. So initially when there's some sort of spread happening, when population density is low, being locally adapted is not favored. And we also have this epistatic negative interaction between the A and the B loci, and all it does is it decreases the competitive ability of any individual to have both A and B local adaptation alleles. So that is the sort of survival selection phase, and then we have a mating phase afterwards, which is random mating, and then we have the dispersal phase, and the generation starts again, going through. And so, how does dispersal work? Well, I set it up so that there's population patches or teams, and they're arranged around some sort of geographic barrier, and Dean 6 is the ancestral contact, and Dean 12 is the terminal contact zone, and you set up ecological gradients. So the terminal contact, local adaptation, the local adaptation alleles have an effect, where if you are locally adapted, you see that benefit. In the ancestral contact, that does not happen. And you also set up allele-specific gradients. So for the A locus, it would be favored on this side of the ring, and on this side of the ring, the B locus is instead favored. And so as a result, you kind of get it so that the A's will tend to give this side on this side. And so the Dean 6 is initially colonized by suboptimal individuals with some sort of standing variation. You can keep it symmetrical, but then we play around with it, we make it asymmetrical. And as a result, colonization kind of proceeds upwards along from Dean 6 along this arm and Dean 6 along this arm as well. And so at equilibrium, you get that the terminal contact and the ancestral contact show different genetic gradients. And so specifically in the ancestral contact, you have it so that suboptimal individuals exist 6, right at the ancestral contact, and you have these heterozygotes that are kind of situated around there. And then what you end up getting is you get this, like for example, in D4 you get the A locally adapted individual D4 or complementary to the D and D8, and it heavily dominates D4. Very similar things in D3. As you travel up the range, you have that sort of thing happening. And then what happens in D12, where you have this very sharp genetic gradient, and it's a mosaic hybrid zone. And any sort of hybrids that are generated here are some sort of suboptimally adapted individuals that have maybe some sort of negative epistatic interaction with their heterozygotes. And when they try and migrate back into D1 or 12, they're selected against heavily due to being competitively inferior. So in short, you've got the gradual ancestral gradient, and you sharp that so that it's not what you think of as a classical ring species, but it is it's an analogous sort of ecologically motivated ring species. And so now I'm going to talk about asymmetry aspects. So you kind of differ what you can think of as the east and the west gradients, or the A and B gradients, because you wouldn't expect nature for them to be exactly the same. So to keep things simple, you keep the B1 fixed, and then you vary the A1. And so when you do this, you get a series of bifurcations. And if you don't know what bifurcation is, essentially what I mean is that you have the contact zone, and it starts to shift even when you do a little bit. That's because this is a sort of population structure or a gene-based model. And then when you continue to do that, and you make it so that now the A has an even smaller benefit than the B in a sort of regional manner, it shifts over. You do more, shifts over. Oh, and if you do more, this ring gets completely dominated. And I should have said this before, but the blue is the B dominated genes, and the red is A dominated genes. And so what exactly does this look like? This is one example of a bifurcation. I don't look at, well, I know it happens at 14.5%, but it actually goes to the contact zones between 3 and 4, but that's a very small sort of parameter space, so you don't I don't care about that too much anyway. So what you get is this D2, it gets fixed for B, so there aren't that many A's around, and in D3 you see that it's about to be kind of like the A is about to be excluded, but since this D4 is dominated by the A local adaptation, it kind of saves the D and allows both of them to be eventually dominated by the A alleles, by the A local adaptation. And if you make that benefit a little bit less, you get complete fixation of the D throughout the ring. So why is this significant? Well, there's this old notion of ring species. They're demonstrations of the temporal aspects of speciation, but it's realized in spatial dimensions, which is great. It made it so that many people study them in the first place, and it works. It's worked for a while, but it's limited in the literature what people think of as an actual ring species and what's worth studying. Because there are probably 20 or 30 candidate rings that don't get as much attention as the sort of classical rings. And so what ends up happening is you end up missing out on a lot of good biology. Specifically, what I think should be a sort of a significant way to look at these complexes going forward is instead ring species as demonstrations of the role that ecological demographic contingency have in speciation. And so what exactly do I mean by that is, well, for one, you have a given genetic architecture, and in one area, due to ecology and demography, you have this sort of gradual genetic gradient that would suggest just a gradual differentiation of forms. Whereas at the other end, you have this very sort of abrupt genetic gradient, which is more reminiscent of speciation. But what's also important is whether speciation could actually happen. I think it's also important to study these ring species because due to asymmetry and to these contingency factors, it's the difference between you having a ring that actually forms in some sort of asymmetrical or relatively symmetrical fashion, like we do see, or whether we actually just say, okay, a ring that is completely swamped and never exists. While kind of finding this would be very hard, finding one of those swamp rings would be very hard. If you find those it would kind of show how likely speciation events are bound to happen based off of these sort of contingent factors. And with that, I would like to thank some people that helped with, well, they kind of were with feedback individuals, and I'll take any comments or questions. Yeah, so there would be acai, I don't know how to say it, but it's seasonal, it's in South Africa. It's got 2 papers about it, but it's been kind of, people are, yeah, and then there's also the slipper spurge, which is around the Caribbean, it's one of the Caribbean islands, and it's differentiation along with different, along with the Caribbean islands due to some sort of local application. There's a recent one that's been done actually in the past little while. It was on some sort of owl, and it was around the Mediterranean, Sea or whatnot, and that was due to local adaptation as well. So kind of that sort of classical notion that kind of gets at these more mutually evolving phenomena, and that's great and all, but it really doesn't allow you to deal with things in a sort of ecological speciation, or even combine